Sloan, and this wonderful, beautiful class is AP Biology, getting ready for Thanksgiving break. They have a hurdle, right? And that's this last exam that we need to take before Thanksgiving break. So this is AP Bio Review, Unit 4, Cell Communication. Okay, so I'll break it down by the different chapters that you have. A couple things while you're looking at this. Let me move me over here. Okay, is one of the chapters, oh, you know what? I might not have put it in this review, and I pre-apologize for that. I'm pretty sure I didn't. I'm pretty confident I didn't. One thing I did not include, I know I didn't, was about homeostasis and feedback. I'm pretty sure I didn't include it. So we'll review that today because I think I forgot to throw it in here. I'm sorry, I apologize. Okay, so this is everything for chapter 9. Do you have this already in your review that I already gave you before? Yes. So you have all of that. I just wanted to remind you of everything you needed in chapter 9 for that. Okay, and basically the reason why it relates to the cell cycle is that you have to have chemicals to communicate when the cell should divide and when it shouldn't divide. Does that make sense? And we can see when we get the wrong signals, we can make too many cells, right? And in that case, what are we dealing with at that point? Cancer, right? And we talked about how there are sometimes cells that are kind of frozen, like if there's been damage to our nervous system, we wish we could turn them on. Yes? Okay, and we are constantly building cells and breaking cells down. That's normally how we maintain what? Homeostasis. Good. All right, so as we look here, knowing the cell cycle and what happens in each part of it, make sure you're comfortable with that. The bulk of the cell spends its time in what? Interphase. An interphase can be divided into 3 steps. G1. If I was going to say what happens during G1 of interphase, you would always tell me what? Growth and what else? Making more organelles. That makes sense to us because we just finished mitosis, right? So our cells are small and we're going to need more organelles and we're going to need to get bigger. But if I'm done, if I don't have enough food, if I'm nutrient deficient, I might say peace out and enter into what at this point? G0. And I'm just going to stop. Do some cells stay permanently in G0? Yes. Okay. But let's say I'm not in G0, I'm still in G1, or I re-enter. What's my next part of that? S. And S stands for what? Synthesis. And what are you synthesizing? DNA. And this is when you make the sister chromatids, right? Do they appear as sister chromatids when we do that? No, because it's just like something that came out of our what? Hairbrush, right? It's all messed up in there. There's DNA and there's proteins, right? Let's talk about those proteins. There were 2 categories of proteins. What's the most common? Not super important, but is important. But what is the most common one? Histone proteins. Histone proteins, remember those spools of DNA and it winds on those spools. You remember that? And you'll have 8 of them together. And what does it form? Starts with an N. Nucleosomes, right? So they're important because structurally it helps you organize your DNA when you're condensing. When are we condensing all of our DNA? Prophase, right? Early prophase. So that helps us organize our DNA so we can clearly see our sister chromatids. There are other types of proteins called what kind of proteins? Non-histone proteins. And we're going to learn in another chapter about their regulatory function, those non-histone proteins. But just some foreshadowing. Do you remember in all of our things about cell communication, how we would see like a second messenger binding to an intracellular receptor and then moving in? Okay, you could be then triggering some of those non-histone proteins to turn on some of the DNA so you can determine what mRNA is there. That would be an example you could relate to. Okay, so that's the S stage. At that point, if I weighed my cell, or let me take that back. If I measured the amount of DNA I had in G1 versus S, what would you tell me? What would you predict to see? Double, right? Because as long as you have the nucleotides available to you, right, and you have the cell signals, what's a signal that would tell me to move into the S stage? Tell me. Cyclin. Specifically S cyclin, right? Would have to hook up with a CDK, which stands for what? Cyclin dependent kinase, which is an enzyme to facilitate that step. But before I go into that S stage, I better make sure everything is working, right? And so do you remember we talked about, what is it called when you stop for just a second and check all of that? Oh yeah, I just gave it to you. Checkpoint, right? And we said that that is a place where P53 really exudes, its impact at that point, because it can do two things. One thing, it can make you stop, so you're checking. And then if you cannot be fixed, what will it do? Take you out. Because we don't want to make more cells that are not correct. Okay? But let's assume we get through the checkpoint. Let's assume we make our DNA and we've got sister chromatids, though we can't see them yet. What happens after the S stage? G2. And in G2, what are we going to be focusing on? Proteins. Because we're going to need a lot of proteins, because we're going to do something pretty major. We're going to go and make another whole cell, right? So then we will move into, once we're done with G2, we'll move into, we'll move into mitosis. Now the M stage, like when you look at its I that gets broken down into G1, S, and G2, the M stage really consists of two things. What two things? Cytokinesis and mitosis. That's what the M stage consists of. Because mitosis is purely the division of the nucleus, and cytokinesis is division of, the rest of the cytoplasm. Okay, so let's go through. The whole thing's called what? Mitosis. Do it with me. The whole thing's called mitosis. What's the first stage? Prophase. The chromosomes. Oh, stop for a second. This chromosome right here, what's holding it together? Thank you. Okay. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules, asters. They meet in the middle at metaphase, away at anaphase, to nuclei at telophase, cytokinesis. Now, if I'm an animal cell, when I do that cytokinesis, I'm forming a what? Cleavage furrow. Can I do that in a plant cell? Because I'm going to first build a cell and turn it into a perfect. Okay? Questions right there you need to ask me. Yes. No. Yes. Oh, G0 is I'm done. I'm old. I'm through. I've done all the divisions I'm going to do. Do you remember when we learned about telomeres, how chromosomes shorten? Okay, and we said one of the things about cancer is that enzyme telomerase keeps the chromosomes a little bit longer. Well, my telomeres have shortened. My time is done. I am moving into G0. That's a normal homeostasis type thing. Yes. Another great question. So you take the organelles you have and you just separate them. However, remember what's going to happen in G1? You're making more organelles, right? Who can make more of themselves? Mitochondria and it looks like what? Binary fission. Because they were once what? Free living prokaryotes. They wait to live in. Now, your endomembrane system, which is basically your ER, both rough and smooth, and your Golgi apparatus, your Golgi bodies, maybe you make some more lysosomes. That would be normal. You can make more of those membranes. You can add to those membranes. The smooth ER will help you, especially with that when you're in G1. Yes.
What's the difference between centrosome and centrioles?
Okay, the centrosome is an area. The centrosome is like a region where the centrioles sit. But do all cells have centrioles? Name one that doesn't. Plants. But they still have a centrosome, an area where microtubules radiate. That is often called the MTOC, microtubule organizing center. Same thing for centrosome. So you have a centrosome and you'll have a centriole sit in it, or initially two centrioles sit in it, right? And then those duplicate and then they go to either side to help you set up your spindle fibers, right? To the spindle that goes across to separate them. Yes.
So you said P53 causes apoptosis or it stops in G0?
First it stop. No, it doesn't have to be G0. Yeah, it's just that always happens in G1. If you do a G1, you're getting checked. Yeah, every time. Because if we fail to do that, we might get an out of control cell. Okay. I mean, because think about it. It's like, how does your body know what cells to make more of and what cells not to, right? I mean, if you think about it, that's incredible because you're made out of trillions of cells and that you know how many you need. Because some cells, like your stem cells, your bone marrow, what comes from your bone marrow? All your blood cells, right? So how do you know how many of those to make? What is the failure of that? Leukemia, right? Any kind of blood cancer, a lymphoma, right? That comes from that. Okay. Anything else on that? Yes. That is one of the examples. Remember when I gave you the 2 cancer genes? So whatever one remember I talked about BRCA1, BRCA2. I talked about RAS. Any of those examples that I give you, you should probably be familiar with them. All right. Oh, I don't have it on there. So let me click here. All right. So then we talked about cyclin dependent kinases, right? And we talked about how do you regulate that cell cycle. And we talked about cyclins, like it could be S-cyclin, like in what we just talked about. As it builds up, it hooks up with those cyclin dependent kinases. Kinases always like to phosphorylate. So when they transfer a phosphate group to another molecule, then it could possibly be turning that molecule on, or it could be turning it what? Off. So that's how cyclin-dependent kinases work. You have various cyclins at different stages of your of mitosis. And that's what you want to know there. If I was going to pay attention to one, I guess I would pay attention to S-cyclin and M-cyclin, right? S-cyclin, you're building up to do the S stage of interphase, and M-cyclin, getting ready to do what? Mitosis. Right? Okay, I'm going to keep going because there's so much in here. Apoptosis, is that normal? Yeah, it's the normal part, right? Okay, and then I talked to you about the P53 gene there. All right, these are just the stages. You have that already. I think you're going to be okay with that, right? So those are just the stages going into metaphase, anaphase, and telophase for your reference. Binary fission only occurs in what? And mitochondria and chloroplasts, right? Prokaryotic cells and mitochondria, and they do not have linear chromosomes, so they can't do that dance of mitosis. They have a single circular chromosome that replicates itself. It basically anchors on. You can kind of see it doing that here and here, and then that cell just divides. That's all you need to know. It is not mitosis, okay? Just in prokaryotic cells. Okay. Next, okay. Normal cells exhibit both anchorage dependence, which except for like blood cells because they're moving around, right? Normal cells have anchorage dependence where they have to have like some basal lamina or whatever they're attached to or another cell, okay? And they also have density dependent inhibition. Like right now we're crowded, yeah? We're crowded in this room. Can any more people come in? No, because of the overcrowding. So normal cells would be like, oh, it's time to stop. For some reason, and we've talked about what those possibilities are, those cells lose both of those things, which means I don't care if I'm crowded and I don't have to stay put. When you don't stay put, that can mean you can metastasize, which means you had your place of origin. If you just stayed right there, a surgeon would be able to go in and just remove it, right? But if it can also lose its anchorage dependence and go somewhere else, it can spread to other places and then disrupt that homeostasis of that tissue or of that organ. And that's when it becomes a real problem. Okay, so here, okay, a neoplasm, just an abnormal growth. You can see they're really piling up right here. They don't care that they are anchored. Now, if they also, they don't care that they are crowded, but if you see them starting to float away, then you can see they've lost anchorage dependency as well. That's just what a cancer cell looks like. Now, or what it does. How a cancer cell looks, They don't have a lot of cytoplasm. What does their nucleus look like? Yeah, it might look very granular. You have abnormal nucleoli, maybe multiple of them, or multiple nuclei. So they look visibly different. Are they specified to their tissue that they're in? Are they differentiated? No. Okay. Trying to see if I put some. Okay. Now, the two main genes we talked about, okay, that are responsible in some way to regulate our cell cycle are proto-oncogenes and tumor suppressor genes. If both of these guys are doing their job, you have no problem. But proto-oncogenes, they are in a position of what? Power. So when they go bad, it impacts everything. And if they go bad, you don't call them proto-oncogenes anymore. What do you call them? Oncogenes. And when you have oncogenes, that means you are not listening to the signals of your body and you're just saying, go, go, go. We need more cells. We need more cells. Now, we know we can recognize when they've gone bad. And what do we want to do to those cells that are not being regulated appropriately? We want to what? We want to kill them. So that's when your tumor suppressor genes come into play. And what they'll do is they'll stop that cell, say, whoa, change your ways. You're not going to change your ways. Then you're dead, right? And we talked about caspases being the one who kind of enact that. If this goes wrong and this goes wrong, this is when you have cancer, right? That's when you have a real problem. You don't have, because this is going to happen, okay? This is going to happen. And some people estimate, remember, that you have a cancer cell every day, and every single day you take care of it. It might be taken care of by a cytotoxic T cell, right? Cytotoxic T cell, you're going to have different antigens because you're not your normal self anymore. You're not your normal cell with your normal surface proteins. You start putting abnormal proteins on your surface, then that's when your cytotoxic T cell says, I don't recognize you, right? And then it's going to what? Kill it, okay? When it's presented on an MHC1, you are done. Exactly. Okay? Did I see a hand up over there in the corner? Sure. They just don't learn. Yes. Yeah, it's just like a chemical, like cytokines. Okay, what else? Yes. Okay, all right. So on a proto-oncogene, if that goes wrong, you're just having too many cells. So boom, that would be cancer, right? But normally those are kept in check by tumor suppressor genes. They don't allow it to happen, right? So if this guy, the assassin, is shooting blanks, we have no way to control our out of control cells, right? And remember the analogy I gave you about like the RAS gene? RAS is like stepping on the gas, right? So if your friend is stepping on the gas and you're like, what are you doing? We're going to an intersection. As long as your other foot can step on you're fine. These are the brakes. But if your brakes go too, if both go wrong, if your brakes fail, you are going to crash in the intersection. Does that make sense? Okay. What else? All right. Yeah. Okay, we're good. All right. And then we go into chapter 33. Okay. Now, before I go into 33, I forgot to put a 31 in here. I don't know why. 31. Remember, the only thing we really learned about in 31, or actually, I hit that in hormones, didn't I? Positive and negative feedback? Yeah, okay. But let's just talk about it right now, just because I want to. Okay? What one is maintaining the status quo, the norm? Negative feedback, right? I control if I have too much of something, and I control if I have what? Too little. And that is most of the feedback in your body to keep things in check. Occasionally, there is situations where we have what? Positive feedback, where there's more, and more. Like if you get a cut in your skin, you've got to make a blood clot. You need to keep getting more and more platelets out there until that ultimately you have a blood clot and it stops. If you are a female and you have a uterus, you're going to keep up. There's positive feedback. You're going to keep up building the uterine lining until it's enough. And then you're going to what? Stop. If you're getting ready to have a baby, you're going to keep up your contractions until the baby comes out and you're going to stop. There aren't a lot of examples about positive feedback. Does it still get you back in homeostasis? Yes. But you go more, gets you more, you more and more until you have some climactic event, and then boom, you're back to normal. That's the difference between those two. When we talked about hormones in relationship to that, we talked about hormones helping you regulate your body, but what does it work in conjunction with? Nervous system, right? Who works faster, nervous system or endocrine system? Nervous system works a lot faster, right? The endocrine system works a little bit slower. And you have things with hormones like where you have contrary hormones. Do you remember that? So we looked at the example of insulin and glucagon, and remember how the insulin would lower our blood sugar and the glucagon would raise our blood sugar to keep it just right. So that was another one. Because I don't think we're going to get through everything, so I'm just trying to tie in where I can tie into things, okay? And then also, because I see we're now here on the endocrine system, it's going to take the immune system, it's going to take a little bit of time right here. Do you remember about the difference between steroid hormones and protein hormones? What do protein hormones do? Can they come in the cell? No, because the cell membrane is going to block them, right? So they bind to which can then trigger what? It can trigger a secondary messenger, right? Remember adenylate cyclase? Maybe it's going to convert cyclic or ATP into cyclic AMP, and that's going to be your secondary messenger. Maybe you're going to open up a channel and let calcium come in, but you are not coming in. Whereas a steroid hormone, because it's a steroid and your cell membrane is a phospholipid, the steroid hormone can go right through, right? And when it goes through, it usually binds, it's still going to bind to a receptor, but it's going to be inside the cell, and we call that a intracellular receptor. And then when it's there, it's usually going to influence your DNA and what proteins are made. Now, when you influence DNA and what proteins you're making, that usually takes a little bit longer. And so that's why we think of steroid hormones as being like our reproductive hormones or long-term stress hormones. Those are steroid hormones. Most of your hormones are protein hormones, and it usually is fairly rapid. Stand at the door and knock, boom, reactions take place through signal transduction, and you get your response. One typical response is to get glycogen to break down into what? Glucose, and then the cell can increase its machinery. Just like the plant hormone who? Gibberellin. Remember that? Okay, that's fun. All right, so I just wanted to, just for fun, I threw that in. Do you have to know gibberellin? No, what's the one hormone you have to know? Auxin. All right, it's okay. Breathe easy, my friends. Okay? Now, whoa, come back. Come back. On your immune system, when we talk about, and are all these reviews I've done on each of these things recorded for you? Yes. Or given to you, right? So you can get those on the YouTube. If there's some part that you don't understand, you can go and get any of the presentations. I've got them in the Google Classroom and Google Stream, right? Okay, on your immune system, the things you want to know is we look at the evolution of the immune system, right? It goes all the way back to like bacteria. Remember we looked at PAMPs and toll-like receptors, right? We looked at what else? What did we look at? It starts with a P. It's unicellular and sometimes multicellular. Do you remember Plasmodium? Do you remember this? What did I teach you? Say it out loud. Oh, yeah, it's the slime mold. Sorry, I was giving you an official name there. All I said was slime mold? Okay. I'm sorry. I'm like, could have sworn I taught that. Okay. Okay, don't stress out. But do you remember the slime molds? And do you remember when they're multicellular? Do you remember the little sentinels and how they would move around? Do you remember that? Okay, there you go. That's all it is. So those are precursors to our immune system. Now, in our immune system, when you look down through, let me see what the next one is. I don't remember what it is. But if we look at our immune system, we had our innate immune response and our what? Adaptive immunity, right? And our innate immune response, everybody has that, right? And what are some of the things involved in our innate immune response? Dendritic cells, which are also known as what? Antigen presenting cells. They kill, destroy, present on an MHC2. I will. Good. So dendritic cells, what else? Macrophages, they eat, and they are also antigen presenting cells. Who's first on the scene, most numerous? Neutrophils. What is a cell that releases histamine for you to at mast cells and basophils release histamine, which makes your blood vessels a little more dilated. So on the arterial end of the capillary bed, what pressure is really strong? Hydrostatic pressure, which is forcing fluid out, and it decreases as you get farther from your heart. What pressure wins out? And that draws fluids back. Do we get all the fluids back in? No. So then what was once blood plasma, and then it was interstitial fluid out and around our cells, then becomes what? Lymph. And that lymph is going to move through what before it gets returned to your circulatory system? Lymph node. And that's where your special forces can hang out, right? And that way, if you're looking for a virgin, you're more likely to find it at a lymph node, right? A virgin B cell or a virgin T cell. And then eventually that gets returned at your subclavian vein. That's right. Subclavian vein and back into your circulatory system. All right. Let's see right here. We did that. Functions of the immune system, you know that. Absorbs excess tissue fluid, absorbs fat at the intestines, produces lymphocytes. We already talked about that. Lymph nodes filter your tissue fluid. What's the partner on that one? Spleen filters your okay. Of your adaptive immune response. You're comfortable with the inflammatory response, right? Okay. No?
Yes.
I fall down, cut myself. Who's gonna come? Platelets to clot it, right? Who's going to be first on the scene? Who's going to show up as smoke? And it's called a good. And it comes in, it starts, and it gets really good. All right, you got that. Dendritic cells are eating, right? You get swelling of fluid because of the histamine, and it's a little leaky, so you get swelling, maybe increased in temperature in order to kill off any bacteria that are there. That's your inflammatory. It swells because more fluid is out there, and that could hit a nerve ending, so that's painful. You okay now? Okay, so that's your inflammatory response. That's your innate response. It doesn't get any better. Never gets any better than that. To really take care of it, you got to get rid of your free floaters and also the ones that are hiding in your body cells. Who's going to take care of our free floaters? B cells. You have that pathogen is going to bind to a B cell receptor. If I'm a B cell that can recognize this virus, would I recognize this virus? No. Why? It's different. Okay. So then what will happen is if you trigger a virgin B cell, is the light green right then? No. You need also a what? T cell who has been presented, I'm not breaking this, but has been presented with one of these on an MHC2 by like a dendritic cell or a macrophage. These two virgins, B and T, now say yes. So the B cell is going to start to clone itself doing what process? Mitosis. So it must have the cyclins to make a whole bunch of itself, right? That B cell has the cyclins to make a whole bunch of itself, not another B cell that wasn't stimulated, right? Okay. And you're going to make a clone army, and you're going to have two categories of cells. What are you going to make most of? Plasma cells. What is the job of a plasma cell? What? And those antibodies are going to be specific for this target, yes? And it'll be great because antibodies have two arms, so they can hang on to two. Anything that's tagged with an antibody is tagged for death, right? And if it's on a virus and it's got an antibody, it cannot gain it can't gain entrance into your body cells anymore, so it kind of neutralizes it, yes? Okay, great. What's the other cell it will make? Memory cells are for a faster response next time. Good. T cells. You stimulate a T cell, a virgin T cell, it'll make how many kinds of cells? 3. What's this one? What's this one? Help. And What's the next one? Memory cells for next time. When you're done waging war on this candle, who will kill themselves? Plasma cells? Cytotoxic, because you can't keep those numbers up, right? And so the only ones left behind are, and maybe a few helper cells. Okay, we'll stick around. Yes? B cells cannot produce antibodies unless they are a virgin B cell that has been given the okay by a virgin T cell, and then they'll become a plasma cell, which will actually secrete the antibodies, or they are a memory cell. And again, it has to come in contact with that pathogen. The reason why memory cells are so much faster is because the virgin needed the whole candle, but a memory cell doesn't need it. Just a little sparkle off of this would do it for me. Okay. What? Yes. Helper T cells amplify and coordinate the response by releasing cytokines. What else? Am I not? Yes, I'm sorry. I'm not sure I understood that question, but cytotoxic T cells are produced by a stimulated T cell, right? So you're either a virgin T cell who's in agreement with a B cell or a memory T cell. And once it's a memory T cell, it could be an MHC1 that presents to it, and it'll I won't have you differentiate between that. Yes.
So when a virgin T cell is choose an MHC1 or an MHC2, does it turn into the cytotoxic T cell?
Oh, I'm okay. No, it's like I was this. I'm going to start doing mitosis, and some of my clones will be memory cells, and some of my clones will differentiate into helper T cells, and some of my clones will become cytotoxic T cells, and then these three versions of me, the original virgin, right? They're going to all wage war. Right? So, I mean, did I become that? Well, I divided in half and then that divided in, right? I just do a bunch of mitosis. Okay. Yes. No, you already made all your virgins. You're done. Yeah. You have what you have. I hope you have a bunch. Right? Because that makes you pre-adapted for the rest of your life. Okay. What else? So cytokines. Can we talk about that for a minute? Okay, cytokines are a way for one cell to communicate with another cell, right? We can do it directly with an MHC1 or an MHC2, some sort of receptor. That's direct contact. But I can talk to you using a cytokine. And there's two cytokines that we focused on. What are the two? Interleukin and interferon. Good. And interferon is when you are infected by this virus, right? And I'm warning all the rest of you, be careful, right? And I'm trying to, you can think of it like I'm trying to interfere with what this virus is trying to do. And so I'm warning all the rest of you, stop doing protein synthesis because you might be making viruses. Maybe you all should just kill yourselves because maybe you already have a virus in it. And I'm also alerting like the helper T cells and other cells, we got a problem. Why don't you all come over here and help me fight this and get this under control? That's what interleukin or interferon does. Interleukin is something secreted by like a macrophage or a helper T cell or a neutrophil. And it's basically like pulling a fire alarm and just calling all the cells, come on over here, we've got a problem. And it amps them all up. If they're eaters, they eat more. If they're killers, they kill more. It just dials everything up a notch. Okay. Yes?
How exactly does it dial it up?
Well, they're going to release a chemical. That chemical is going to have, you're going to have a receptor for it if you're a cytotoxic T cell. That's going to trigger a signal transduction to increase the speed at which you're doing whatever you're doing, killing. Yeah. All right. Next, We already talked about the inflammatory response. We've talked about T and B cells. We're good on all of that. You have all of these in your presentation. The summary, interleukin, interferon. Oh, chapter 26. Okay, chapter 26 we just did, right? Chapter 26 we just did, so that you probably feel pretty comfortable about. We hit that hard. So let me just jump into hormones a little bit more to make sure you understand that, right? Hormones are when a gland secretes a chemical into your what? Bloodstream. And it targets organs that are at a what? Distance. At a distance within your body. If I target another whole organism outside of me, like somebody I'm trying to mate with, that's called a pheromone. Okay. And the reason why they work, let me just finish the sentence. The reason why they work is you can flood your body with that hormone, but different cells in your body have different what? Receptors for that, right? Okay, and then you have the other reviews. I encourage you to study, but make sure you sleep tonight too. I know you have math and history and Chinese and trifold boards.
I remember.